So it's my pleasure today to introduce today's Grand Round speaker, Dr. Brittney Harrington. Dr. Harrington received both her Bachelor of Science and PhD from the University of Queensland in Australia, where she began her research into ovarian cancer. She then came to the United States, where she was a postdoctoral fellow in the NIH National Cancer Institute in the Laboratory of Cellular Oncology, where she continued her research into ovarian cancer. Since 2019, she's been a postdoctoral fellow in the Women's Malignancy branch at NIH NCI. And over the years, she's received many awards for her research, as well as her teaching and mentoring students. Her research focus on ovarian cancer spans many areas, including NF-kappa B signaling, immunoconjugates for detection and inhibition of ovarian cancer, glycogen utilization, and targeting tumor-initiating cells.
Recording in progress.
I met Dr. Harrington in person for the first time last week at the American Association of Cancer Research Ovarian Cancer Conference. So I'm looking forward to hearing more about her research. The title of her talk today is Using Translational Genomics to Target Ovarian Cancer Stem Cells. So Dr. Harrington, if you can figure out how to share your screen, we'll be set.
Okay.
Yeah, I will. So part of the Zoom issues this morning were that the Zoom app wouldn't open, and so I had to join by the desktop, the web app. So
Okay.
I think the only way I can share is by sharing my screen. So you'll see the Zoom screen itself before you see the PowerPoint, I think.
That is fine. Thank you.
Here we go. Oh, no, look, it's working. Okay.
Okay.
Well, hopefully this goes better than yesterday.
Thank you.
Yesterday, I did a practice with my mentor, Dr. Annunziata, and over Microsoft Teams, and the whole thing shut down. My computer crashed and all of that. So let's hope the computer tantrum was only yesterday and not today. Okay. I think, yeah, I can't see the chat or anything, so I'll just go ahead. And you can let me know if anything happens, like the sound changes or anything like that.
Yep, looks good now.
Great. Okay. So yeah, thank you for the really kind and introduction, Amy. And I will talk about my research, what I've been doing at the NCI, where I've been investigating ovarian cancer stem cells that we've been aiming to target these to prevent disease relapse. And we took the translational genomics approach to do this. If my screen will advance. There we go. So briefly, I'll introduce ovarian cancer, its incidence, its treatment, and some information about its disease progression, as well as what I mean by cancer stem cells, which I might default to saying tumor-initiating cells, TICs, and let me get my laser pointer as well. And I'll go through the research aims that I had for this project at the NCI, where I've been trying to identify drugs to specifically target the tumor-initiating cell population, and as well as find new targets by identifying genes that are essential to the tumor-initiating cells. And through this, I found the gene UGDH as a essential gene for tumor-initiating cells. And then beyond its functions in the tumor-initiating cell population, I found that it had important roles in the tumor microenvironment of ovarian cancer. So I'm excited to share that too. And to align these research aims with the objectives of today's talk, so the objectives were to define translational genomics, and I've defined this as the integration of both functional and discovery-based approaches using genomic methods. And this was used to in research aims 1 and 2. Objective 2, to compare models of ovarian cancer disease progression is, really it's all 3 of these. And finally, to identify drugs and essential genes to target ovarian cancer stem cells to prevent disease relapse. So a little bit about ovarian cancer, its incidence, and so on. If you're unfamiliar, it is diagnosed in approximately 20,000 women per year in the US, and at a median age of 63. So this is the generally postmenopausal population. And the lifetime risk of the general population is about 1 in 78. But this risk can be higher if there's a genetic background or hereditary BRCA mutation and other such genetic risk. And it's the most lethal gynecologic malignancy and the 5th most common cause of cancer-related death in women. And this is partially because most women that are diagnosed with the disease are diagnosed at an advanced stage. And this is partially because of the symptoms of ovarian cancer can be very hard to detect. And a lot of these symptoms can be vague or explained by other things, or a lot of these symptoms are also similar to menopausal symptoms. So often we're seeing diagnosis at an advanced stage. And for treatment of ovarian cancer at whatever stage, it is a combination of surgery and chemotherapy. And just to highlight again, the stage really makes a difference in survival. So if stage 1 and 2, early stage diagnosis, it's a more confined disease. You get a much higher survival rate, 5-year survival rate. However, most patients coming in at stage 3 or stage 4 have a really decreased survival rate. So in terms of treatment, as I said, surgery combined with chemotherapy, and this can happen 2 ways. Initially, if the patient is a good candidate for complete surgical resection, no macroscopic disease, they'll undergo surgery first, followed by chemotherapy for 6 cycles, and as well as a maintenance drug for 1 to 2 years or however long until they progress. However, there's also the option for chemotherapy up front for 3 cycles of the same, followed by surgery if there's widespread disease or lots of tumor nodules, to shrink the disease before surgery, and then followed by the other 3 cycles of chemotherapy. So they both get 6. And initially, there's a good response to chemotherapy in most patients. Most patients will initially respond to platinum, and the chemotherapy drugs most commonly used are a platinum drug, either carboplatin or cisplatin, combined with a taxane, which is paclitaxel or docetaxel. And these work in combination because carboplatin, or the platinum drugs, platinum can intercalate with the DNA inside the tumor cells and can cause DNA damage, which usually would be repaired in healthy cells. But when you combine DNA damage and paclitaxel, DNA damage needs to be repaired in S phase of the cell cycle. So paclitaxel or taxane drugs, they inhibit cell cycle progression. So DNA is not repaired and the cells undergo mitotic catastrophe and they're no longer viable. So in terms of relapse, the treatment changes a little bit depending on how long before they relapse. So if it's more than 6 months since their last chemotherapy cycle, they can be re-challenged with carboplatin because they're seen as a responder, and they can have another chemotherapy drug other than a taxane, and they can also add bevacizumab, an anti-angiogenic therapy, for another 6 cycles as a maintenance therapy. However, if it's been less than 6 months since their last chemotherapy cycle and they progress, then they won't be re-challenged with platinum because they didn't really respond, and another chemotherapy drug can be chosen. Bevacizumab can be given again, but ultimately these patients will progress. And so this is the area of my interest, is once patients come to relapse, can we prevent relapse after their initial response so that they don't relapse and undergo this progression? So another factor I wanted to touch on in the ovarian cancer is the tumor microenvironment. So this is a busy slide. I apologize. I took this figure from a paper, but I really just wanted to highlight that there's multiple factors within the ovarian cancer tumor microenvironment, not just the bulk tumor or the tumor cells, the cancer cells, immune cells, and stromal cells all communicate and exert this pro-tumor effect. So what's unique, I guess, about ovarian cancer is its metastases. Rather than spreading via the blood or lymph nodes like a lot of other cancers, the ovarian cancer metastases are really local. They don't generally extend beyond the peritoneum. You don't get really distant metastases. And this is aided by the accumulation of ascites fluid, which can facilitate the survival and movement of cancer cells that are shed from the primary tumor into other organs in the peritoneum. The tumor microenvironment is also highly immunosuppressive. So there's actually quite a lot of infiltration of particularly macrophages in the ovarian microenvironment, but these are not the pro-inflammatory anti-tumor M1 type. They're generally the M2 immunosuppressive type. And as well, there's an active stromal component. So you've got the cancer cells, immune cells that are immunosuppressive, they're co-opted. You also have the stromal cells such as cancer-associated fibroblasts, endothelial cells, mesothelial cells that also enhance the tumor. And I just wanted to add to this figure tumor innovation. It's something that's becoming really important in the field. And the AACR special conference last week, or the week before, we heard about this from Ronnie Dratkin and Paola Vermeer. They're showing that actually nerve cells, nerve bundles, are not just a passive part of the tumor microenvironment. They're actually promote tumor as well. So they're in the blue down here in the pro-tumor effect. So when I'm talking about cancer stem cells and relapse, that is the thought that in ovarian cancer, there is survival of chemo-resistant stem-like tumor-initiating cells, clones, that can regrow the tumor. So if you're not familiar with the cancer stem cell hypothesis or theory, within the bulk tumor, there's a mixed population of these so-called cancer stem cells, or TICs, I might call them, that can proliferate and regrow more cancer stem cells, but they can also grow and fuel non-cancer stem cells, proliferating cells within the bulk tumor. And so in this mixed population, when treated with chemotherapy, the non-cancer stem cells, they're more sensitive and the tumor will shrink. These cells trying to divide, they undergo mitotic catastrophe, and so you get tumor shrinkage. However, any resistant clones, the cancer stem cells, will be resistant to chemotherapy, and they can regrow the tumor. So this is the population that I'm trying to target, and the TICs, or cancer stem cells, they can be identified by the expression of both intracellular and extracellular markers. So extracellularly, these cluster of differentiation molecules, particularly CD133, they're expressed on the cell surface, so we can detect them by flow cytometry or using antibodies. And then intracellularly, this enzyme, aldehyde dehydrogenase, ALDH, has been shown to have not just important features as a marker of these cancer stem cells, so if they have high ALDH activity, but also they promote chemoresistance through drug efflux. And I will be talking about this ALDH today, and other features or functions that promote chemoresistance I added to this, etc., include oxidative stress response and metabolic plasticity to really be able to avoid cell death by chemotherapy. And so to study the tumor-initiating cells, we have used very defined culture conditions to enrich for this populations. So we use a 3D culture model in vitro to be able to enrich this population. And these are a non-adherent spheroid culture. When we culture these cells in this way, we can see that they express all the stem markers, they're chemoresistant and aggressive in vivo. So this is a flow cytometry plot of both CD133 expression on this axis and ALDH activity. And in normal adherent conditions, this cell line does not express either of these markers very highly. And when you put them into the 3D spheroid culture, we see increase of this population. And importantly, again, when we culture in traditional adherent conditions, they have a much lower, they're much more sensitive to carboplatin, for instance, compared to the 3D cells, as reflected in the IC50. You get a huge, you have to have much more drug to be able to kill these cells. And then in vivo, if we inject the same number of tumor cells, depending on their culture condition, you'll get larger tumors in a shorter time frame from the exact same amount of cells. They are aggressive and regrow tumors in vivo. So for me and for my project, I was interested in targeting the TICs to prevent relapse. And so I was thinking about how do we specifically target them? Do they have specific vulnerabilities compared to the non-TIC cells? And what genes, there must be more targets within these TICs, what's making them special compared to the non-TICs. So the genes that promote survival. And so I was comparing this traditional adherent culture, which cancer stem cells may be in there, but we're really comparing this primary proliferating population grown in 2D adherent conditions to the 3D tumor-initiating cell enriching spheroid conditions. And using a translational genomics approaches, we integrated a number of methods. So I initially used a high throughput drug screen to identify drugs that may be targeting these population, combined with RNA sequencing that we did, comparing, again, these two populations. And in a different approach, whole genome siRNA screen as well, which I'll talk about later. But essentially, this is what the approach looks like. So you have RNA-seq. That's your genomic approach. and then a drug screen or a viability screen using whole genome siRNA. These are our functional approaches. So this really gives a way more targeted TIC-specific candidate where a drug, it might work on the 3D cancer cells, but if it doesn't make sense, if it doesn't make sense with the gene expression changes, then it's not going to be a really viable drug. Similarly, I might identify a gene that's really important or interesting for cancer stem cells, but again, if it doesn't make sense expression-wise, then it won't be a good candidate. So first, I will talk about the high-throughput drug screen. So we use this high-throughput drug screen in collaboration with the NCATS Institute to screen almost 2,000 drugs to identify what was going to be sensitive for TICs, or what would make TICs more sensitive compared to adherent cells. And overall, we actually found that drugs that could augment oxidative stress were effective against spheroids. And when we combined our high-throughput drug screen findings with the RNA-seq findings, we saw this similar enrichment for genes involved in drug metabolism and oxidative stress response in the spheroids. So this really filtered down and strengthened our hits from this screen. So almost 2,000 compounds were screened against both cells grown adherently or under the TIC enriching conditions. We were interested in which genes would affect the TIC condition grown cells rather than the adherent. And so from that 2,000 genes, this is zoomed in here, we found that we actually examined 4 drugs here, disulfiram, bidoxylene methyl, olescamol, and seleninomycin, because we thought it would be interesting if it could affect both the TICs as well as the adherently grown cells, because maybe we can use that in combination with chemotherapy rather than a relapse drug. And this drug here, it's an IAP inhibitor, which someone else in the lab was simultaneously investigating, so we left that one alone. And these drugs, a platinum drug, doxorubicin, they are known to be anti-proliferative, so it makes sense that they were more effective against the adherent cells, but it also shows that the tumor-initiating cells are resistant to these. So a little background on these candidate drugs that we identified from the screen. Disulfiram, firstly. It's also known as antabuse because it was traditionally used as an drug to treat alcoholism. And it does this by inhibiting ALDH, the enzyme that is also a marker of cancer stem cells. And its function in the cancer stem cells is it promotes drug metabolism. So chemotherapy comes in and ALDH high activity really just flushes it right back out. So this is a good candidate. Bidoxylene methyl, we were initially interested in this because it has been linked to the NF-kappa B pathway, which is something that the lab traditionally has worked on in ovarian cancer. But we found it was included because of actually mainly its anti-inflammatory properties, and it can regulate the antioxidant response, which we found oxidative stress is really important in the TICs. Olescamol is an interesting one. It causes just excessive amounts of oxidative stress. It produces a lot of reactive oxygen species. However, it's not really fully elucidated what it actually does molecularly, what it targets, where it goes, that kind of thing. And it produces these reactive oxygen species by disrupting the electron transport chain. So it's sort of a mitochondrial drug, but it's not really fully known. And finally, solenoamicin. This one came out, and in the literature, it's been shown to inhibit cancer stem cells of other cancers. So that was a good hit for us. But additionally, multiple mechanisms have been reported for solenoamicin, including inhibition of oxidative phosphorylation. So this also contributes to oxidative stress. And NRF2, I didn't mention before, is an important regulator of oxidative stress. So both solenoamicin and butoxylene methyl are thought to inhibit or activate NRF2 to promote antioxidant response. So we initially, the screen was for whether these drugs could inhibit viability. So the first thing we did was make sure that they could inhibit spheroid viability in multiple cell lines, making sure it wasn't just a cell line effect or an artifact of the screen. But we also investigated these functional properties that we know are important for cancer stem cells, whether they could form spheres, whether they could produce the stem markers, importantly ALDH, whether they could withstand oxidative stress, and a new assay that we call in vitro relapse. And so when I've been talking about adherent culture versus TIC culture, I'm talking about spheroid growth, because we put them in these non-adherent conditions and they become a spheroid, and it makes them really resistant to chemotherapy, and they resemble what you see in ascites fluid from patients. So initially we tested out, so I guess spoiler alert, these two drugs were the most effective, but we did try all of 4 of the drugs, but disulfiram and olescamol had the biggest and strongest effect. So we tested, again, adherent versus spheroid culture. So these were already formed spheroids. Could these drugs penetrate those spheroids and have activity? The short answer is yes, particularly disulfiram. You can see that they are much more sensitive, the spheroids, than the adherently grown cells at these concentrations in multiple cell lines. And olescamol, it doesn't look so good here, but in the other cell lines, it was making these spheres really sensitive. So we validated that from the screen, so we kept going with these drugs. Next, we also wanted to make sure or see if the cancer stem cells were able to regrow a spheroid if they've been exposed to this drug. So we measured them over 6 days, and we added the drugs when we plated the cells into these spheroid growth conditions. And as you can see, they all form spheres pretty effectively at no drug, but there's a dose-dependent decrease in sphere formation for all of the drugs. But you can see disulfiram and olescamol had a much stronger effect at lower doses. And so, again, more stem cell functional markers, markers of stemness. So we examined the expression of CD133 and on the cell surface, as well as ALDH activity intracellularly by flow cytometry. And we found that, again, disulfiram, olescamol, they were the most effective at reducing these populations. The double positive population. And when we were thinking about how oxidative stress was involved in the cancer stem cell phenotype, we looked at reactive oxygen species levels, as well as mitochondrial function. But we found that that the cancer stem cells are actually really sensitive to reactive oxygen species. If they can't control the levels of these of ROS intracellularly, they're really vulnerable. And so elastomol really hugely increased the reactive oxygen species within the cancer cells, which was shown by previous literature. But actually disulfiram also increased the reactive oxygen species, which we were a little surprised by. And here we have hydrogen peroxide. It's a control for increased reactive oxygen species, and elastomol is getting kind of close to that control wild level of ROS. And so as I mentioned, an in vitro relapse model. So this is a model that we developed to have an in vitro model of relapse compared to an in vivo model, which we also have. But as a final preclinical or in vitro step. So this is where we take the adherently grown cells. Every drug condition, all the cells are grown just adherently in the presence of carboplatin for 48 hours. And so we take the drugs, the cells that are still viable after exposure to carboplatin, which should enrich for the tumor initiating cells, and then we put them into the enriching culture and either treat them again with PBS as a control or repeat carboplatin and compare that to treatment with the candidate drugs to model the chemo-resistant phenotype, see what we could achieve with that. And so what we found was increased cell death when we treated with the candidate drugs compared to a second round of carboplatin. So this is the amount of cell death measured by annexin-5 and propydium iodide, double positive staining. And obviously, if you treat with PBS, the cells are happy. But treating spheroids with carboplatin, it doesn't make that much difference. Carboplatin first with PBS or carboplatin followed by carboplatin, again, there's not that much difference. They're less happy about it, but sure. But then when you go and try these candidate drugs out in vitro relapse, they've had carboplatin and they've regrown. Then they're treated with disulfiram or elastomol, and they really increase their cell death, even above the dual carboplatin treatment. So these two drugs had the highest efficiency. We were really excited about it. I said that. However, when we came to try elastomol in vivo, we found that it had really high toxicity, completely unacceptable for animal work. And actually, elastomol was also trialed in an ovarian cancer or peritoneal cancer as a therapy, and the trial was stopped again for cytotoxicity. So unfortunately, it's a very toxic drug against cancer stem cells, but it's not going to be clinically viable. So we examined disulfiram and solitomycin as our kind of second choices. And so we used an in vivo relapse model. So this is a relapse model where we treat the mice with surgery and chemotherapy similar to patients and give these new drugs following that chemotherapy to prevent relapse. And so we inject the ovarian cancer cells into the intrabursal site and allow them to establish for about 2 weeks. So then the ovaries are removed, so they have an ovarectomy, recover, and then they're treated with 3 cycles of carboplatin and paclitaxel. All the mice get that treatment, and then they either get vehicle and a vehicle control to relapse, or we tried our drugs, clindamycin and disulfiram, for three cycles and watched their survival. And strikingly, we saw that both disulfiram and clindamycin worked well as maintenance drugs. So in the gray bar, the mice have had actually no carboplatin paclitaxel. They've just been given PBS, and they have very short survival. But when we do add the carboplatin paclitaxel treatment, they do increase their survival. So they are responding to chemotherapy, but they are relapsing. So I find this very exciting for this model. And then, as you can see, the added drug treatments, disulfiram, clindamycin, extend out that survival much more. So particularly we were interested in disulfiram because of it's able to inhibit ALDH. And what we found was ALDH is a more broad ALDH inhibitor. And so with the NCATS Institute, we investigated whether we could get really specific inhibitors of ALDH to see if that would give us a stronger effect, more potent against the tumor-initiating cells. And so specifically, we were looking at the ALDH1A1 enzyme inhibitors, and they could reduce ALDH activity as expected. So this is just going off the activity of ALDH. Disulfiram here very strongly inhibits the activity of ALDH, but these specific inhibitors, they also reduce the activity, but these are again specific to ALDH1A1, whereas disulfiram can target many. And we compared them against all of these functions, the sphere formation, spheroid viability, in vitro relapse, and found still disulfiram is kind of unbeatable compared to the specific ALDH1A1 inhibitors. They still had good activity, but not as good as disulfiram. And similarly, they just really didn't perform how we thought they would. Looking at these viability curves, again, disulfiram really hits the tumor-initiating cells in orange, whereas the other two drugs had close effects on both adherent or tumor-initiating cell populations. And so we wanted to dive into this a bit more, and we know that other researchers in the ovarian cancer field have shown ALDH has good activity, disulfiram has good activity. It's a broader ALDH inhibitor. And because we saw, disulfiram can even inhibit spheroids that don't express a lot of ALDH. If disulfiram is only inhibiting ALDH, or all the ALDH inhibitors, even cell lines that don't really have much going on, they still are sensitive. So we found that disulfiram is actually acting by inhibiting ALDH, but also disrupts classical NF-kappa B signaling, which is the REL-A P65 pathway. Again, we come from a NF-kappa B lab, so we investigated this. And so what we found when we compared disulfiram to the other inhibitors was that the P65, the active NF-kappa B classical pathway signaling, was able to be decreased by disulfiram treatment, and it never came back up to the control level. And when we added inhibition of the pathway, so we inhibit the P65, so the activity of the pathway, not just the expression of the pathway, disulfiram became ineffective. So disulfiram has to inhibit the classical NF-kappa B signaling for it to have this effect. So we're continuing these studies of disulfiram, actually in combination with other drugs, and looking at its metabolic phenotype, which is really interesting. And that's that publication will come out the next few months, led by Dr. Kei Kudo in the lab. And so coming back to the research questions and using the translational genomics approaches, I want to talk about my second project that focused on UGDH. And so this is where we used the RNA-seq data again to look at gene expression changes of adherent and tumor-initiating cells and combine this with findings of a whole genome siRNA screen. And so for this, the siRNA screen took whole genome worth of siRNAs, up to 3 siRNAs per gene. So it ended up being tens of thousands of genes that were inhibited by siRNA. And we did this both on adherent cells or spheroid cells. And the readout for this, the functional aspect, is cell viability. So if you inhibit this random gene in an adherent cell, does it kill the cells? And same for spheroids. And that's fine, but we wanted to specifically target these spheroid cells. And so we plotted the Z-score to see the difference between genes that inhibited adherent cells we weren't so interested in, but genes that could specifically inhibit spheroid cells. And so of the thousands of genes, I just plotted the top 5,000 here and really investigated this top 20 that could specifically inhibit the spheroids. And so from this screen, we identified here the top 20, so those red dots on the graph. And apologies if you can hear any background noise. There's a garbage collector. So again, the Z-score, we see the highly negative score is meaning that it specifically inhibits the spheroids. And what I chose to investigate is UGDH, which I'll get into, but it wasn't the top just based on this screen. And I do want to also mention this NAMPT inhibitor, NAMPT enzyme is also what Dr. Kei Kudo is working on. So it came out in this screen as well. So it's a good candidate. And so taking our functional data and comparing it using our genomic data, we had to see if this was going to be a viable target. And plotting here the fold change in expression. So on this end of the axis, these genes were more highly expressed in adherent cells, and these genes on this axis are more highly expressed on spheroids, and comparing the p-value, so how significantly different are they. This cut out a lot of the genes from this list, and I highlighted a couple here that we were still interested in. UGDH is here, so maybe the expression isn't that different, but in terms of function, it was more significant. So this kind of highlights, if I chose just one screen or the other, UGDH I wouldn't have investigated. And through some more literature search and using this RNA-seq data to find the hallmarks that are really overrepresented in the spheroid population, and looking at expression of RNA and protein using the human protein atlas, UGDH became my #1 target. And so a little bit about this protein. It's an enzyme. It's UDP glucose dehydrogenase, UGDH. It has been studied in a couple of cancers for its pro-tumor functions, such as particularly glioblastoma and lung cancer, but a whole host of others. And really at the start of this project, one paper came out in ovarian cancer, which I was annoyed by, but I've characterized the expression in ovarian cancer, so I'm not so annoyed anymore. But its function, briefly, is it takes this substrate, UDP glucose, from glucose, and hydrolyzes it to become UDP glucuronic acid, which is a building block for all of these pathways, particularly hyaluronic acid, which is up in the cell membrane. It has a lot of functions in signaling functions in ovarian cancer. It's also used for proteoglycan synthesis, glycosaminoglycans, so post-translational modification, as well as glucuronidation, which is a drug eliminating pathway, as well as some other functions listed in the literature, which I'm not going to go into, as I said. So first, I wanted to characterize its expression in ovarian cancer because it had not been really examined apart from a couple of cases here and there. And so I was lucky to have access to a few tissue microarrays to really analyze this. And I had my PhD supervisor, Dr. John Hooper. He had a high-grade serous 96 patient tissue microarray with matched primary and metastases cores from the same patient. I also looked at the other subtypes of ovarian cancer, of epithelial ovarian cancer, so clear cell, endometrioid, and mucinous subtypes, which were provided by the Australian Ovarian Cancer Study, which is David Botel's group. And interestingly, and what I will highlight, is there was also access to these molecular subtypes of ovarian cancer, of high-grade cancers, also from the Australian Ovarian Cancer Study. These were initially characterized in 2008 by Tothill, and I will go into them later. But first, the primary and metastatic high-grade serous cancer. Really what was striking was how highly expressed UGDH was. 97% of the cases, high-grade serous cases, unclassified, they were all, basically all highly expressing. We really got hardly any negative in either primary tumors or metastases. And I did also split the staining, the scoring of the staining, into nuclear localization or cytoplasmic, because other papers, particularly in lung cancer, showed a prognostic effect where nuclear UGDH was associated with a poorer prognosis and so on. But when I looked at primary or metastases, I didn't really see a big change in the localization or expression. They were both really high. And so this made the survival analysis challenging or a bit confounding, because I really didn't have that many negative cases, negative or low, but I did every comparison. I did negative and low versus moderate and high. I did every comparison, but survival phenotype didn't really come out, or progression-free survival based on expression or localization. But what I did find was that comparing, particularly in the cytoplasmic staining, patients, so in terms of survival, at the end of the study, 25 patients were still alive, and their average survival time was 2,000 days, say. Low cytoplasmic expression, most of the patients that were alive had low cytoplasmic expression. Compared to the patients that were deceased at the end of the study, deceased from this cancer, 52 of these patients, most of them, 48 of them, had high UGDH cytoplasmic expression. So it didn't pan out to survival analysis or progression-free survival, but it's an interesting, I guess, feature. So looking at the other subtypes, the clear cell, endometrioid, and mucinous subtypes, I had only 10 of clear cell, 18 of endometrioid, 24 of mucinous, and here's just a nice picture of that, of the typical kind of staining I got. And when we quantified the amount of staining, we saw clear cell had moderate expression, much like this one case. Endometrioid had lower expression overall of UGDH, which is also interesting. But mucinous by far had the strongest staining of the lot. I also did all of the survival analysis and comparisons, but mainly because I didn't have the numbers. Clear cell was interesting in terms of survival and progression, but 10 patients wasn't enough to get a statistically significant phenotype. But that's something I would love to continue to look at. So finally, the molecular subtypes that I mentioned. Initially, back in 2008, there was whole genome sequencing, exome sequencing, and gene expression profiling of high-grade cancers. And so to develop a molecular signature to see if we could separate out based on genes and whether this related to function. And so 285, both serous and endometrioid cancers were examined from this original paper, and using clustering analysis, they developed 4 subtypes. This has been reproduced by other studies multiple times. And so these are designated in the literature. It's either for mesenchymal, for instance, either the C1 subtype or mesenchymal. But in this presentation and in my paper, I use both designations to be extra clear. And so the importance of these subtypes was not just, okay, we can cluster them differently. They also have a functional effect in terms of the survival and relapse in these patients. So the mesenchymal subtype, C1, had the poorest progression-free survival and overall survival compared to all other subtypes. And these were matched patients, so it's really something in the mesenchymal subtype that's driving this this poor survival. And so when I looked at just high-grade serous cancers, I found there was not any prognostic differences, nothing clear. But when I used this tissue microarray specifically looking at the molecular subtypes, the C1 mesenchymal subtype, which had the poorest survival anyway, also had the strongest expression of UGDH, and high UGDH was much more, they had a much poorer survival. And on the opposite side, this differentiated, it's kind of functionally the opposite of the mesenchymal group. I saw the opposite phenotype where low UGDH in this group had a poor survival effect, which was honestly frustrating when I first found it out, because I thought, how am I going to explain this? How am I going to look at this? And so in order to study this, we classified ovarian cancer cell lines into each of these groups that I color-coded to keep the same. And so I chose to study the mesenchymal group, I chose OV90 cells, and to study the differentiated group, chose these ACI23s. And so in order to investigate UGDH-specific functions, I knocked down UGDH with shRNA, so expression goes down. And for the differentiated, I wanted to increase the expression to see if that higher expression of UGDH in this phenotype would correlate to a better prognosis. And so we looked at all of the stemness markers again, to really validate what we found in the initial screening, and found that indeed, if you inhibit UGDH, spheroid growth is affected but not adherent. So that's perfect. It matches the screen in the C1 mesenchymal cells. But in the ACI23s, when we overexpress, we actually get better spheroid growth. And this is okay. I mean, I expected this. But importantly, it doesn't affect adherent cells in either condition. And then looking at more functional assays, sphere formation and stemness markers, they were both inhibited by UGDH, which is interesting because here we get overexpression of UGDH and it actually decreases the amount of these stem-like cells, although we get more spheroids. And so because I was interested in the molecular subtypes and how different they are phenotypically in patients, the C1 mesenchymal has a lot of stroma, and the differentiated is a much more quiet tumor. I looked at these both in vivo. So I looked at first whether they would change the growth of tumors, and yes, if you knock down UGDH in the C1 mesenchymal group, the tumors just really don't grow, and they have much more significant survival. But in the C4 differentiated group, we would expect the low UGDH would have a worse survival, and by increasing UGDH in this group, their survival could be extended. But I actually didn't see this. I didn't see a significant difference in tumor growth, although this trends that way, and the survival, it didn't look different. So I wanted to look at the microenvironment, the stromal activity in both of these groups, and I looked at the tumors. So using this Mason's trichome stain, I found that when you overexpress UGDH, these tumors generate tons and tons of collagen. And this might have contributed to the size difference. So if we compared actual tumor size without the collagen, it may show us a difference in size here, because they really just pumped out the collagen. And because I thought maybe this is just a mouse thing, and how could this be happening, I wanted to test if it was a secreted factor. So overexpressing UGDH, was it a secreted factor that's causing this collagen abundance? I know I'm getting close to time, but I think this is my last slide. So when we took just the medium, UGDH overexpressing cells, whatever they were secreting into the media, it caused fibroblasts that we cultured to increase their collagen abundance. So this is something I'm working on now, taking mass spectrometry to find out what this difference is. So to summarize quickly, we took this translational genomics approach to integrate screens and produce focus targets to prevent disease relapse. And we found through this that TICs have different vulnerabilities, specifically to drugs that can affect oxidative stress. And when I investigated UGDH, interestingly, I wouldn't have found any of this data without both screens coming together and without looking at the various molecular subtypes of ovarian cancer. And I'm continuing my work on UGDH in the future. And so thank you to all the collaborators and the getting the tissue for this project. And there's many people to thank, but and thank you for inviting me to speak. So I'll stop my presentation.
Okay, for those of you going to the quality meeting at 9, you can stamp off. Amy, I've given you host privileges. You can stick on with our guests. Thanks.
Okay, great. Thank you.
Recording stopped.
Yeah, I was going to say a lot of them have to go to this other seminar.
Yeah.
Oh,
Yeah, no worries.
did you want to ask a question, Andy, before?
I did. I'm actually not even going to that one. I'm going to a different one
Oh.
on RAM and breast cancer.
Okay.
But Brittney, that was a great talk. And I've worked with Amy before. I've worked with others in our gynonc program, including Boris Winterhoff, and ovarian cancer details. I have two quick questions, hopefully. They may not be quick, but I'll be a little late to the other seminar. One, I was wondering, thinking honestly about tumor initiating cells, cancer stem cells, and the CD133 ALDH positive subset. How plastic is that? In your model systems, can you get a sense of how adept a cell line or a group of cells, a heterogeneous group of cells in culture can be in producing or replenishing that fraction?
I guess I haven't really done that, but Carrie House, she was a postdoc in the lab that she did all of the culture to enrich, and she optimized the conditions. And these conditions with the specific growth factors and all of that really enriched it's particularly ALDH activity is most important. CD133 expression does fluctuate between cell lines and passages, but ALDH activity is really what we found.
Okay.
Was stable.
The reason I was asking is, I mean, that part of your talk made me think back to stuff that was a long time ago. I mean, a long time ago. Gil Smith, I mean, some of the work that Sean Morrison's done with tumor initiating cells and melanoma, and, again, serial transplantation through mice, and the fact that you can get down to, at least in some of Sean's experiments, like 1 in 4 cells can have this capability. And again, it just depends on how plastic and how able cells are to kind of change state, particularly in vivo. The other one that I thought was really interesting with the UGDH, getting all my alphabet soup mixed up.
I know.
But that expression, again, some of the stuff that's come out of the University of Minnesota, thinking about the mesenchymal subtype, is really that phenotype, at least in the gene expression studies, is largely driven by the stroma itself. But I thought it was particularly striking that UGDH and that it looked, at least from the power of images you were showing, that expression is pretty much confined to the tumor cells. But I was going to ask, do you see its expression in cancer-associated fibroblasts or tumor-associated macrophages in that microenvironment either?
We did, for the immunohistochemistry, I asked for a stromal mask. So I'm only showing the epithelial, the tumor cells expression.
Okay.
I'd have to go back to really dissect out like which had strong stromal expression, but I think it was very few. It was really clean staining, and it just didn't.
Yeah, it's interesting crosstalk between the tumor and the stroma in that condition when we think about what the biology of the mesenchymal subtype is. So I thought that was cool.
And that's why I did the conditioned media experiment as well, to be really like only expression confined to tumor cells. Yeah, so it's
very
interesting.
cool. Yeah, great talk. Thank you very much. Thanks, Amy.
You.
Great. We have 2 other people have their hands up. So if you still have your hand up, I can't really see your name. Oh, here we are. Okay.
Yeah, I need to dash into the other room in a second too. So, but I wanted to really ask this question quickly. So thank you very much, Dr. Harrington. So quick question. What induces the UGDP? When it's there, we know what it does, but why is it induced? Is it like constitutively expressed in the tumor stem cells, or does something have to induce it to produce its effect? And as a follow-up to that, do you think hypoxia has any role in the induction of some of these tumors? And I don't have time to tell you why I'm interested in hypoxia, but just a quick question.
Well, I can answer the hypoxia one quickly first. UGDH can be actually inhibited by HIF1 alpha. So I haven't really studied that because I was interested in the activation of UGDH. So there is something there, but I just haven't got into it. And then how is UGDH activated? It is constitutively active. It's really a metabolizing drug, an enzyme, and it has highest activity in the liver, but in normal ovarian tissue, it's not really expressed. And it's only ovarian cancer or the other like lung cancers where we get really increased expression. I don't know what the promoter is for its expression. There's a couple of promoters that have been identified as being important for UGDH, but really I think it's protein and its expression and its activity that's important.
Yeah, because
Does that answer your question?
Yeah, that it does. It does. I was just curious about that. So if we know what it does, and if it's not necessarily good for the stem cells, I'm just wondering maybe another level of approaching treatment for this cancer to actually prevent its induction at some point.
Yeah, unfortunately, there's no like specific inhibitor for UGDH. There's some broader ones, but they don't. Yeah.
Thank you very much.
Things. Thanks so much. Thanks for your question. Oh, Amy, you're muted.
The one hand up with is Saichev still there?
Hi there. Yes. Good morning. Great presentation. That was fantastic. One question is I have a couple of questions related to reactive oxygen species. So I know you measure percent of ROS of reactive oxygen species. Do you know what species was the most dominant? And then that was just an overall measurement of the oxidation state.
No, it was just an overall measurement. Yeah.
Yeah. And have you thought about, because the major homeostasis mechanism to maintain reactive oxygen species is like NADPH, right? It's like the reducing power of the cell. So have you measured to see like how to compensate for that in terms of a cytotoxicity, or maybe add NAC, which is N-acetylcysteine, to kind of regulate that process and kind of still, you know, because I know it seems like that's the Achilles tendon of the cells of the TICs, they're reactive species, but like if you go through that over like this kind of like creating that environment, but in a suboptimal conditions, not like at the excessive level, right? Like, and then still get those TICs, because it seems like that is the weakness.
Yes, yeah. I haven't been doing that, but Kay Kudo in the lab, he's really focused in on the NAD, and he's doing, well, his paper will come out soon, but the enzyme he's really interested in is the NAMPT, which is a niacinamide. I can't remember the rest of it, but it's an NAD converting enzyme. It's involved in redox. So his work, really shows that with the TICs, they're just so sensitive. And if we can target something like that in that pathway, rather than just dumping a whole lot of ROS on these cells, maybe that's going to be a better approach. Yeah, because
like the main regulators is like NRF2, right, which is kind of like negative regulated by KEAP1, and so that reactive oxygen species what disengage NRF2 and turns on the scavenger system. So like it'll be interesting to see how in your system this NRF2 itself is regulated. Most likely you touch on NF-kappa B, but that's like a major hub. So if you knock that one down, you knock the whole system down. So have you done any NRF2 studies?
We didn't inhibit NRF2 to see if the drugs had changes in their effects, but we did look at NRF2 localization with like nuclear localization with each of the drugs. And so we saw pretty much all of the drugs except for bardoxolone, which was interesting. They all caused NRF2 to shuttle into the nucleus. So it was active, but something was preventing NRF2 from actually carrying out its detox function. So yeah, we didn't go much more into that. And I mean, the pandemic happened, so we didn't get to, but we published the paper, and
Great work. Thank you.
Oh, thank you.
Great. Okay, we got Jim here with his hand raised, so
yes,
so go for it.
Dr. Harrington, thank you. That was a terrific talk.
Thank you.
So my own research is in the area of hyaluronan, chondroitin sulfates, and how tumors progress and multiple tumor types. I would just have a comment. The study that I'm familiar with is from the TAMIs, and they're in Finland, and they study breast cancer and hyaluronan metabolism, and they've identified that the substrates of these enzymes, or the products of these enzymes, excuse me, of UDP, the alphabet soup again, are increased, and that is not only UDP glucuronic acid, but also N-acetylglucosamine, obviously important for synthesis of extracellular matrix glycosaminoglycans, but also generally in metabolism. So I guess one of the comment is just that what you may be, they didn't look at the enzyme itself, and what you may be onto here is a more general change in tumors that reflects either their change in metabolism, the tumor initiating cells, and how they need to change the metabolism of glucose in order to survive. So I just, like I said, a terrific talk, and it may be broader than ovarian. Certainly it's going to be important in ovarian, but something to think about. And I'm happy to give you their names. They're they're in Finland. As somebody in the Hyaluronian field that might be worth hooking up with in terms of
definitely
expanding what you want to ask for questions. I just, like I said, I think it's terrific. So thank you very much.
Well, what I didn't show was I have been looking at metabolism and the metabolites, particularly actually UDP glucose, not the product, and finding some really interesting features there. And I think it's when the cancer cells are switching more to this glycolytic phenotype,
right,
there's more UDP glucose being produced. And I'm trying to see if it's really going through the pathway and producing more hyaluronan, or if it's being excreted, which is really exciting.
Absolutely. And I just one additional quick comment.
No, I appreciate it. And
so the question is, are these, so one of the things that we think about, we study a proteoglycan called chondroitin sulfate proteoglycan 4. So it's got a unique core protein associated with it. It's on the cell surface. But those studies have led us to think about the idea that tumor initiating cells are obviously very important. But another way that recurrence can happen is by cells invading into the stroma and actually not being accessible to chemotherapeutic agents, and they can give rise to recurrence that way. So this chondroitin sulfate that we have has very similar effects that what you're talking about in terms of increasing the resistance to cisplatin, promotes their invasion, promotes their growth, promotes mesenchymal transition by mechanisms that are not entirely clear. So the question for you then specifically is, are your tumor initiating cells, are they invasive? If you can culture them in adherent culture and see what they do, but if you ever look, after you do that, can you dissociate them and ask whether they are more invasive, and could they live in tissues where they can come back again, like in recurrence, like you're talking about? Is that?
Yeah, I haven't done that with UGDH, but with
or just with the cells in general, just with the tumor initiating cells with all the various markers, are they more invasive, and could they, another way that they could recur would be to essentially escape the environment where the chemotherapeutic agent is?
Generally the spheroids, yes, they are more invasive. I don't know that we've dissociated the spheroids and put them as single cells.
Or we allow the spheroids to attach to a matrix and see what happens in terms of their.
They do invade. They do invade, that, but we just haven't separated out the cells individually to see. But that would be interesting.
Your topic is near and dear to my heart. I appreciate it. Thank you.
That's great.
I'll be happy to send you, if you want. Amy, you can give me Dr. Harrington's email. I can send you the paper by the TAMIs, hyaluronan gurus in our world. So
yeah, that would be fantastic. Thank you.
Okay, great. Thank you so much.
Yeah.
No more hands up. I'd like to ask a couple of questions. First, if Jim's still on the call, I would like to say that he was one of the people that actually got me going on doing the spheroid experiments way back in the day and saying, nobody's looking at them. Nobody's looking at them. So, I mean, we did do experiments like with cell lines and make spheroids and literally and then take patient ascites cells and saying, do they invade do they stick, do they spread, do they, et cetera, and then taking mesothelial cell lines, coating plates to mimic the peritoneal cavity, and seeing that only like 1 in 10 of these spheroids from a patient would dissociate. And at the time, nobody ever heard of tumor initiating cells and such. And so, looking back at it now, when we still do spheroid experiments, we're saying those probably were, the 1 in 10 were the tumor initiating cells that were within that spheroid, but from the patients. But my questions, I had a couple more that are more simplistic instead of blowing my own horn or crediting them with my whole career. But the idea was, whenever you use the OV90 cell line, I was just wondering, how did you choose those? Because we're really having issues with getting a cell line, an ovarian cancer human cell line that grows quickly enough for you to do the experiments and then to be nicely spheroid forming and to do everything. And we have not used the OV90 cells. And I was wondering, why did you choose those to begin with?
Initially, so the drug screen was the very first thing. And before I arrived in the lab, they were testing out the growth properties of, they tried OV90s and I think OVCAR3s, OVCAR8s, PEO1s to see which one would be best for the drug screen, like even the platform growing the spheres and all of that. So OV90s were the best for that. And then I guess at the same time, the RNA-seq was being done using the OV90s as well, because Carrie House in the lab, she was interested in NF-kappa B, and the OV90s, they really have their classical and non-classical NF-kappa B signaling, so she was also interested in them. So it kind of was serendipitous that both used the OV90 cells, but I mean, I really like them. They express a lot of UGDH, so that's another reason I wanted to use them. And they form spheres really, really well. They're aggressive, hard to use in vivo, but yeah.
That's good. Okay. And then the other question was, whenever you show experiments where you're saying, now we're going to be measuring like CD133 in the surface of adherent versus spheroid, for us to do flow cytometry, we can't be doing spheroids. And I'm just wondering, do you dissociate them? And then is there concern that you're cleaving off some of the CD133s or the cell surface things, or do you just use a mild trypsin or we've tried a couple of dissociating things, and if the spheroids are super tight, like we get from patients, there's no way they'll come apart. So I was just wondering if you had some tricks of the trade there for spheroids and flow cytometry kind of things.
Yeah, we do dissociate them. The cell lines, sometimes you can just get away with the EDTA based, and really like filtering them through these like 70 micron filters to kind of get them apart. But for tighter spheres, we've used Accutase as well.
Yeah, we've tried that too. Yep.
Yeah. Okay. And, there's another one that someone else is using, but yeah, really I've only used the cell lines, and we can gently dissociate them.
Okay. And have you tried doing any of these experiments with real live patient cells?
No, I wrote a grant to be able to do that, so I'm hoping.
Okay. Yeah, I mean, that'd be cool. I mean, we've been saving patients' ascites fluid, the fluid, and we've saved the cells cryopreserved so that we could do experiments like this. And our protein is Nectin-4 that we're more interested in. And then now, we've used cell lines for a long time. We're doing OVCAR5s. And now we're saying, oh, well, let's try patient samples. And they seem to be just very hard to pull apart for types of experiments like that you're doing in these things. So that's good. And what was my other quick question? Everybody wanted to go. Yeah, so like the viability assays for spheroids, that was another question. So we've been trying to do experiments in the IncuSite machine. I don't know if you have such a thing where you watch, it takes these little videos over time and time lapse photography, and so we can watch the spheroids forming, and we've used well plates that have wells in them that are more conically shaped than flat bottom things. But sort of the problem is that when we want to look at viability of those cells and say, if we add this drug or that drug for the screening or whatever, those spheroids, it's hard to tell whether they're dead or alive because they're in the spheroid formation. So I just wondered if you had any tips in that regard too. I hate to be asking all these questions. I don't know if anyone else has any more questions. So
yeah, I'm sorry for going a little over time. So for the viability, we use the cell titer glow assay. So we really use that as our measure, and it just measures the whole well. But Kay Kudo, he's been using the IncuSite with the that live dead stain that fluoresces green or red, and he's been getting some good data out of that. But I can't remember. I think he was using the V bottom wells.
Right. Yeah.
Found them better than the conical or the flat ones. But for when I did the sphere formation, and initially I was sitting down drawing like around every single sphere and counting them. But I, with the microscopy core, I developed this assay to be able to, using basically AI, to count the spheres with a stain a viability stain. So that made it much easier.
Yeah, because it seems like, since it's a spheroid, you can't really tell if it's just cells on the surface that are dying or just ones on the inside, or what gets counted. And so we were kind of worried about that so if this tighter glow is the thing that you've used and it seems to work to get into the spheroids.
It's from Promega. We used, so there's just the original or classical cell titer glow, but there's also a 3D glow. So it's for spheroids as well.
From Promega? Okay. All right. I hope Kristen's still on this call because she's the one in the lab who's doing some of these experiments and is like, oh, what are we going to do? Okay. Well, it's like you published this in 2020. So I thought, maybe things have improved over time. So if people in your lab are using the IncuSite, I think the problem is we have the IncuSite 5 and it only has a limited number of measurements it can take or colors. And so using the red, I think it does orange, but it doesn't do red. And so to use that thing that goes from, they're live and they're green and then they turn red, we couldn't use that. And then we were worried about, we started even trying GFP labeled cells and saying, but once they form those spheroids, there's such a big glob. If you use those, the round bottom wells, the way it takes pictures. So anyway, we were going to try some other ways and give up on the camera and go back to the old counting stuff. So that's my question about that. And
also, we use the ultra low attachment plates, and so they're flat bottom. For our spheroids, so we don't have to use the round or conical bottom.
And I think we ran into a problem again with that IncuSite using non-adherent ones. Is like they don't approve. They have their list which ones are
focus and
and that was one that they didn't approve of any of them. And so anyway, we need to make sure that we have the latest software, I think, because maybe they will use some of them.
I found the microscope using that live imaging, just a regular, would say regular microscope, but live imaging microscope, getting all of the images every well at 10x even, and using the AI to train the AI and it counts the viable cells. That's been a lifesaver. It's like,
Okay.
I've been kind of using it as like a plate reader.
Right. Yeah.
It's a bit.
That's what we got to do. Okay. Does anybody else have any questions? Because I can let you go. I'm not seeing any hands raised because I have this limited new Zoom. Okay. Well, thanks so much. This was really great. And I saw we had like over 70 people watching you. So that's really great. And I know that not everybody in my department is an ovarian cancer researcher. So it's good to get the introduction. And there were a lot of residents and fellows also who, don't really know much about the whole thing. And so it's good you showed some staining.
Yes,
like to see that.
I thought about the pathologists. Yeah.
Okay. Well, thanks a lot. And I guess we'll see you at the next ovarian cancer meeting.
Yeah.
Okay.
Thanks very much.
Okay.
Thanks again, Amy. All right.
Bye-bye.
Bye.